Hello and welcome everyone. You've reached the CyVerse webinar series. I'm Tina Lee, CyVerse's User Engagement Officer, and today's webinar is on next generation sequencing analysis with emojis and CyVerse, presented by Ray Enke of James Madison University in Virginia. As many of you know, CyVerse is a cyber infrastructure project funded by the US National Science Foundation, and our free webinar series helps fulfill a key part of CyVerse's mission, which is to train scientists on how to use CyVerse's computational resources. I'm going to cover some housekeeping really quickly before we start the webinar. Ray's presentation is approximately 30 to 40 minutes. There's a demo in it, so stay tuned for that. Please keep your audio muted and open your chat window where you can type questions for Dr. Enke. He has graciously agreed to allow viewers to ask questions during his presentation. So you may unmute to ask him your question, or if you type your question into chat, I'll read that into the video recording. We will be putting this video recording and other materials and posting those onto our website later today for you to review at any time. So in addition to these webinars, CyVerse offers focused workshops on open science skills and container technology. Please visit our website learning pages for more information about those teaching and learning resources. We've also recently launched the Discovery Environment 2.0. It's a brand new interface with all the same great functionality plus more features and functionality to make your collaborative research even better. Please do check that out. And now I'm pleased to introduce our presenter, Dr. Ray Enke, an associate professor at James Madison University. Ray teaches both undergraduate and graduate level courses in genetics and cell and molecular biology. His own research centers around epigenetic regulation in retinal neurons. And early on, Ray recognized that, quote, students who can compute and manage data are the researchers who will be making the discoveries that lie over the next technological and biological hill. So as a leader in developing effective learning materials in applied genomics and bioinformatics, Ray's work really helps CyVerse achieve our mission of training the next generation of scientists. So thank you so much, Ray, and welcome.
Thank you, Tina. Thank you so much for inviting me to give this webinar. And I should also thank you and CyVerse. I'm a huge CyVerse user. JMU, we have incorporated a lot of CyVerse tools. And so it really means a lot to me to be able to come here and share some of the things we've done with CyVerse tools. And my webinar today is titled NextGen Sequencing Analysis with Emoji and CyVerse. And what I'll do is I'll sort of break this into three parts. First, I'll give sort of a general overview of some of the different CyVerse tools that we're using at JMU. And we also have 3 summer workshop announcements that I'd like to tell you all about. Then I'll tell you a little bit about this specific implementation of an intro level coding exercise that we've put into the undergraduate curriculum that focuses on next generation sequencing, yes, using emoji. And then lastly, I'll give a demo of this activity using these unique new tools that CyVerse has made available called VICE applications, which are really, really cool, and they make integration of bioinformatics in the classroom really, really seamless. And yes, please feel free to throw questions in the chat or just chime in. So first, a little context about JMU. We're sort of a large public undergraduate university in Northwest Virginia, Harrisonburg, Virginia, about 20,000 undergraduate students, about 1,200 biology majors, and about 40 full-time biology faculty. And over the past 6 or 7 years, we've had a lot of administrative support to revamp our curriculum, particularly our lab curriculum, to have an emphasis on course-based research. And we've been using the AAAS Vision and Change guidelines for that curriculum development. And what's ended up happening is we've adopted a lot of CyVerse tools because, again, they're so awesome and they're so easy to use, both for students and instructors that may not have a lot of background experience. So probably our most successful implementation to date has been the complete sort of revamping of our first year biology experience. We have these 2 freshman level courses that students take in the fall and spring semester during their freshman year. We call it Bio 140 Foundations and Bio 1 Foundations and Bio 2. And we completely restructured these classes, lecture and lab, kind of updated the content But the lab portion, we decided that we were going to incorporate a course-based undergraduate research experience using DNA barcoding. So this is a large enrollment course. About 1,800 students a year go through this course. That is 100% of our majors, and then a large handful of non-majors, so chemistry majors, other life science majors. So, we have these great big lectures, but students will break out into their weekly lab that caps at 24 students. And they do this really unique DNA barcoding workflow that we've worked with our collaborators at the Cold Spring Harbor DNA Learning Center. They've really developed the pipeline for this workflow that fits really well. And we've sort of developed it for freshman level students. It's an extremely engaging cure where students go out into the field, they collect a specimen, it could be a plant, fungi, invertebrate animal. They bring it back into the lab where they do some molecular analysis, DNA extractions, PCR. Here's a photo of 3 students Instagramming the bands on their gel that they're very excited about. The caption says, "If you can't tell, we are gel electrophoresis masters." And this sort of culminates at the end where they do bioinformatics analysis of their Sanger sequencing reads that they send off. We get the Sanger sequencing data back, give it to the students, and they put it into this sequencing analysis software called DNA Subway Blueline that's been developed by educators and programmers at the DNA Learning Center. And this is an extremely user-friendly interface for doing research-grade bioinformatics analysis, sequence viewing and trimming, blasting your sequence to find out what it is, sequence alignments, building phylogenetic trees. These are, all of these research grade softwares are packaged into this user-friendly interface. It's very easy for students to run. It's very easy for students to generate data. And so we're giving them a high dose of this in their freshman year in these 2 semesters. So we've published this methodology in CourseSource a couple of years ago. This paper is describing the first semester, our 14-week lab experience. So if you're interested in DNA barcoding, I would highly recommend this. It's a great sort of entry level, trying to get your students to do research grade bioinformatics, but anyone can do it. It's even no matter if you're dealing with novice students or novice educators, anybody can learn these pipelines and kind of pull them into their classes. And I'll highly recommend our supporting materials. We have 80 something supporting materials down to PowerPoint slides that you show at the beginning of lab, the lab activities for students, the instructor versions, prep notes. We have everything there so that if you wanted to do the entire course the way we did it, or just bits and pieces of the course. So that is there for anybody to use. And again, I'd highly recommend it. The DNA Subway software that the DNA Learning Center has put together, it goes beyond just this blue line for Sanger sequencing analysis. There's all sorts of other softwares in this interface as well for annotating gene sequence. The green line is for eukaryotic RNA-seq analysis. And I'm really excited about this new line that's been developed, the purple line. This is for DNA metabarcoding analysis. So rather than Sanger sequence analysis, this is with next generation sequence analysis where you can do 12S microbiome analysis, or 16S microbiome analysis or 12S environmental DNA analysis. And for the past 2 summers, we've run DNA barcoding workshops using the barcoding workflow with Blueline. We're going to be running another one of these workshops this summer that we're looking for participants. I'm also excited to announce that we're also running a metabarcoding workshop this summer. So we're looking for participants, and I'll throw the link up in the chat at the end of the talk. But if you're interested in DNA Subway, it's a subsidiary of CyVerse. I don't know if I'm really explaining that right, but it's a CyVerse entity that is great for entry-level students. So I'd highly recommend the DNA Subway tools. And here's the workshop information. I'll flash this up again at the end if anybody's interested in hearing more about workshops. Okay, but back to the sort of the big picture here is one of the fallouts of incorporating this 2-semester cure into the freshman year. This is sort of a snapshot of the rest of our 4-year curriculum. We have a couple of more core classes that all of our students take, and then they all sort of branch off and they can take all of these different upper level electives. But having the barcoding in the first year really allows us to ask the questions, how can we teach, where in the curriculum can we now teach more advanced computational biology concepts, and how do we do that effectively? So previously, one of the major problems we would have is students wouldn't even do introductory bioinformatics probably until their junior or senior years if they were in maybe one of these advanced classes like genomics, and maybe that's only 20 students. But now all the students are getting a baseline in bioinformatics. So we now have a real opportunity here to kind of step it up and use more advanced tools, preferably still in the core during their second year. And then, also how to teach these concepts effectively. So when thinking about sort of advanced computational biology, a lot of times biologists are thinking about next generation sequencing technology. These, of course, are the contemporary high throughput sequencing techniques that are really just pervasive in every branch of biology at this point. Whether you're an ecologist, a microbiologist, or a molecular biologist, chances are you are dealing with data sets that are produced by these sequencers, or you're minimally sort of reading the literature of experiments that are done on these next generation sequencers. So I think this is, there's a great opportunity to teach students what next gen data looks like, how to analyze next gen data, but also for educators to think about what are the challenges to analyzing and teaching next gen analysis. So my collaborator, Jason Williams, and his colleagues put together this really great survey study a couple of years ago, asking the question, what are the barriers to integrating bioinformatics into undergraduate education? And what they found was a lot of the same, and this was surveying many educators across the United States. And what they found was sort of the same several issues kept cropping up again and again and again. So educator issues such as lack of expertise and training in the subject, overly full curriculum. If I'm going to teach a 4 or 5 week RNA-seq analysis, what am I taking out of the curriculum? It's not always an easy thing to do. Lack of vetted teaching materials. And then on the student side, students need to have some prerequisite knowledge to do like an RNA-seq pipeline. They'd have to know something about RNA, and they have to know something about the transcriptome. And the big one I've found is lack of student interest. You can sit a student in front of a computer and tell them they're doing biology, but that doesn't mean they're necessarily going to enjoy it or remember anything. You have to make these exercises engaging, and that's a tall order because it's very different from picking up a pipette and running a gel. So all of these issues really resonated with me, and I started thinking about ways that I can maybe adapt some of these pipelines to take into the classroom, particularly into the core classroom. So early on in the undergraduate experience. So I teach an upper level genomics class, and that's a 400 level class, small group of maybe 20 students. They've signed up, they've elected to be in a genomics class. So those, that type of student is kind of game for, okay, I'm ready to dig in and do 4 weeks of RNA-seq analysis. That's a different story when you're talking about a first semester sophomore who's taking a genetics lab. That would be a lot to say, hey, we're going to do 4 weeks of RNA-seq analysis, even though maybe you haven't learned about RNA expression yet or transcription or any of that stuff. So one thing that I've tried to start thinking about is how can I deconstruct these pipelines, and maybe instead of kind of forcing students all the way through a next-gen sequencing analysis pipeline, what are some bits that I can break off and take into the lab, especially early, a core lab, like a 200 level genetics lab. And one of the things I have sort of honed in on, and I should say this approach works great. So whatever sequencing methods you're into, whether it's metagenomics, RNA-seq, or genome assembly, you can break off little bits and do, for read alignment or sequence assembly. You could just take these little pieces into the classroom and make little engaging activities. One that I've been zeroing in on is this very first step of pretty much any next-gen sequencing analysis pipeline. Again, it doesn't matter what application you're doing, genome assembly, RNA-seq analysis, that's the 2 workflows I'm showing here. Very different workflows, but the first step is the same. No matter what you're doing with a next-gen experiment, the first thing you have to do, you have to check the quality of the sequences that come off the sequencer. That's a every pipeline requires that, because you want to make sure that the data you're analyzing is high quality data. If you're using poor quality data, you're going to get inaccurate and non-replicable results. So this is a good opportunity, I thought, to focus in on this early step in next-gen sequencing analysis, to talk to students about, what does the data look like? A FASTQ file. This is the fundamental format of next-gen sequencing data. We can actually take a look at, here is what one single read in a FASTQ file looks like. So there's 4 lines to this read, the read identifier, the actual sequence, the first base call is a G, the second position's an A, et cetera, et cetera. And for each base call in the read, there's something called a Phred score that is a predictor of the probability, the error probability of that base call. And this is a symbol that equates to a number between 1 and 50. And here's the Phred quality score telling you something about the base call accuracy. So essentially, the sequencer is going to output a base at every position. It's going to say, I think this is a G, and then it gives you a Phred score for every base call to say, I'm 99% sure that it's a G, or I'm 99.9% sure it's a G. So this is also a good opportunity to talk to students that, a Phred score of 20, for instance, gives you a 99% accuracy. Students would probably feel pretty good if they got a 99% on their exam. But in the world of sequencing, that's actually considered low quality, right? Because when you're talking about sequencing millions or billions of bases, you've got to have something more along the lines of 99.9% accuracy. Again, depending on the method you're using. But at least for, say, Illumina, which is one of the main sequencing techniques that's currently used, 99.9 is kind of the expectation. So a Phred score of about 28 or higher. Keeping in mind, this is just one sequence in a FASTQ file that may have 20 million reads in it. So then you can talk to students about what is this FASTQ file? It's essentially thousands or millions of these reads that have sequence and Phred scores. It's not something that you can open. It's a text file, but it's a huge text file. It's not something that you can open in Microsoft Word. It's not something that's meant to be read by a human. It's machine readable, right? You need software to open up and look at these massive files to make sense of them. So that leads to, how do we assess the quality of this type of data? And then we begin talking about softwares that are used in the field. For example, FastQC is one of the popular softwares in the field. You feed your FASTQ files into the FastQC software. The QC stands for quality control. And it goes through a 12-point pass-fail quality metric where you generate plots, kind of like this one here, a per-base sequence assessment, where you have Phred score on the y-axis, so 0 to 38. Phred scores above 28 are in this green zone of high quality. You have this beige intermediate zone, and a Phred score below 20, or 20 or below, so 99% or below accuracy, it's considered low quality. So if you look across here, and I should say the x-axis is the position of the read. So what you have is an average Phred score for all of the reads in your FASTQ file at each individual base. So what we're seeing here is that the first base position, let's say there's 20 million reads in our file, the average Phred score of all the 20 million reads at this first position is very high, and you can see that's the same going across. So this is an example of high quality data. But your data could also look like this. This is something where the FASTQ file starts out looking good, but then the data dips down into something that's not probably usable. And I should say it's salvageable, but you would need to do some work here. And what you would need to do if you were actually using this data is you would need to apply some filtering or trimming software to take out the bad reads or the bad bases in your reads. And again, just kind of hammering home the concept that analysis of low quality data will yield inaccurate and misleading results. So one approach to doing this, and again, this is more of like an upper level approach for students who are really vested in learning kind of complicated bioinformatics. So we do this activity using CyVerse discovery environment in my upper level genomics class. I have a link right here to the class activity. This is something where, I give students a bunch of FASTQ files, I share with them. They use this app in the CyVerse discovery environment called FastQC, the software I just talked about. And students can open the app, they can feed in the FASTQ files, and they can generate all of these research grade tables and plots that tell them about the data. So I was really interested in taking an activity like this and bringing it down to a sophomore level high enrollment genetics lab that is not just going to be taught by me, but it's going to be taught by a bunch of other faculty who have very, varying degrees of expertise, from absolutely none to a little bit with genomics and bioinformatics. So this, this right here, what I'm showing you, can maybe be a little boring or tedious to intro-level students, and also maybe a little bit scary to instructors that don't have background in next-gen sequencing. So I asked myself the question, how can I teach next-gen sequencing analysis to intro-level undergraduate students in the most engaging and fun way possible? And that was sort of the genesis of this fun emoji activity. And so I have to give a shout to my collaborator, Andrew Lonsdale. He is the person that developed the software. I love the backstory. He was working on his thesis research. Well, I should say he should have been working on his thesis research. And developing this FastQE software was kind of like his side project while he was ignoring his thesis research. And the way this came about is I just saw this come across my Twitter feed one day, analyze next-gen sequencing data with emoji. And that grabbed me. I was like, that's really cool. And my second thought was, this would be a lot of fun to bring to class. Students will really grab onto this. Emoji is like a cultural thing that will resonate with them much more than these plots, right? So this was the idea, was to use FastQE, and you can probably imply here the name FastQE, it sounds like FastQC, so it does the same thing as the FastQC software, but instead of outputting those boring histograms, it outputs emoji. So if you have high quality reads, you get emoji that look like these. If you have low quality reads, you get these type of emojis. So it's a fun and intuitive way for students to understand the QC step of next-gen sequencing analysis. So kind of taking an important, but maybe a little tedious aspect of next-gen sequencing analysis and kind of making it a bit more fun. The other hook that really grabbed me with this is not a point and click tool. This is a command line tool where you have to script everything to run FastQE. And so that was another thing that I was really searching for. I wanted to find a very sort of light intro level scripting activity that I can bring to a 200 level class. As I can say, there's a real shortage of scripting in our current biology program. So we're looking for more ways to incorporate that into the curriculum. And I think a lot of undergraduate biology programs are like that as well. So I'll tell you a little bit about the program that Andrew developed, and then I'll just give a quick demo of it. So again, here is a read in a FastQ file. It's individual base calls and then associated Phred score. And remember, I told you each of these symbols is associated with a number between 1 and 50. And when you read a FastQ file into a program like FastQC, it equates that symbol with that number. So what Andrew did when he designed his program, it's a very simple idea. He equated this symbol, rather than to a number, to an emoji. So if you have these two columns over here of Phred scores that are associated with over 20, you get all of these sort of different versions of happy emoji. Whereas if you have a Phred score of 20 or below, it outputs an emoji that is not so happy or alarming. So it's, emoji output is indicating the Phred quality score of each base. So we teach students you just need some very simple scripting commands on the command line to run this program. So we really walked them through this. We realized that this is going to be the first scripting experience for the vast majority of our students. They freak out anyway when they first see it. So do the instructors, by the way, but this is an activity for novices. Anybody can do this. So they're doing really simple commands like cd for changing directory, unzip for unzipping compressed files, ls for listing out your directory, and finally installing and running the software on the FastQ files that we give them. And when they run the FastQ file with FastQE, this is the output. And you might be saying, well, what is that? Well, what it is it's the exact same thing as this FastQC per base quality plot. It's just outputting it with emoji. So we still have an average Phred score at each base position for all of the sequences in the FastQ file. And so where you're seeing the reads start out high quality here and they dip down, we can see the same thing here. Except the students who have absolutely no background in sequencing analysis, they're able to look at this and understand the quality of the sequencing file, of the FastQ file, way more than they are. They're just intuitively, they can understand this better than this. But when you look at this, you start out with high quality bases, and then toward the end, you have all these alert signs, poop, frowny faces, and the worst of all, a crying kitty. Okay, so, and you might also be saying, well, this is silly. This is not a research grade tool. And that's exactly right. FastQE is a fun, engaging tool, but students did have to learn a few simple scripts to make this happen. And they struggle with this part. And once they get the emoji up on the screen, there's this real sort of aha, like, oh, I did that. I made those emoji come up using these scripting commands. And then what we have them do is they pivot from using FastQE to running another piece of command line software, something called FastP. And this is an actual research grade tool. It's a filtering and trimming tool where you feed in your FastQ files that need to have some bad, poor quality bases trimmed out. Again, with just a few simple commands, students will take this FastQ file that they saw with their emoji output that needs some trimming and filtering. They run this through FastP using a few simple commands. And this time they're generating actual research grade outputs. So a table of pre and post data, their pre and post per sequence base plots. So you can see here is the pre-filtering where we start out high and then the quality gets low. Here's after filtering where all of the reads are now really high quality. And then we bring it back to the emoji, of course, at the end. They rerun their file one more time through FastQE. Here's their original file they generated that has some low quality at the end. After they've run it through the FastP software, they run it back again and they could see that they've now eliminated all of the low quality bases. And so they've actually, they've gone from not having any or very little scripting experience to actually doing real research grade analysis on the command line in a matter of 60 to 90 minutes. So it's a really fun activity. We've implemented this now over the past, I want to say 3 semesters, and we published this method recently, another CourseSource publication. We really like CourseSource. And this is an activity that is designed to fit, it's a one-off activity designed to fit into a 60 to 90 minute class period. So, kind of, again, we were trying to think about these barriers. Like we wanted something that got the students' interest, was easy enough for educators and students to learn, didn't necessarily need a lot of time. You didn't have to take a lot out of the curriculum to put it in there. So it kind of checks a lot of boxes, something we were looking for. When we did some assessment of our students, we asked them a couple of questions. The first was, do you have any prior experience writing computer scripts? Again, keeping in mind, this is a 200 level course. The vast majority of the students in here are sophomores. And the data shows us that this is almost always I should say a lot of times this is their first experience writing any scripts. And I see that as a really, great opportunity to give students, biology students or other life science majors, sort of their first introduction to coding and scripting, doing something fun, but also doing something that's actually analyzing next-gen sequencing analysis. This other question we asked them, are you interested in conducting future scripting analysis? Trying to gauge like if they liked it or if they hated it. And I guess there's different ways. This is in the eye of the beholder. I'll tell you the way I see it. You can see that there's a lot of dots that kind of span into these negative responses, intermediate responses, and positive responses. What I see here is that there's a lot of students that did not hate it, and there's even some students that liked it. So what we're doing is we're offering up an early coding experience in a biology class that a lot of students are enjoying or kind of indifferent to, and the hope is that maybe somewhere later in the curriculum where they're encountering coding, scripting, programming in another class, they've already encountered it once, and possibly it'll be something that kind of grabs their interest in the future. Okay, so like I said, we implemented this. It worked really well, but we wanted to find, so one of the main issues we were having is that students were doing this command line analysis, and they're using this machine or this machine, some Mac users, some PC users. I actually had a major issue that we didn't figure out until I brought this into the classroom the first time, is that emoji are not supported in Windows. So I'm a Mac user. I didn't realize this. So me and my students who are Mac users were able to zip through this exercise no problem. all of my PC students were like, this doesn't work at all, what's happening? And also, anytime we're doing any kind of bioinformatics or computational stuff in the classroom, the biggest hurdle is user-specific problems that have to do mainly with their machine, what's loaded in their machine, what's not loaded in their machine. And that's where CyVerse comes in and can be a huge help, especially with these new apps that they've developed called VICE apps that are, they're called Visual and Interactive Computing Environments. There was a whole CyVerse webinar devoted to explaining what these VICE apps are a couple of years ago. I'd recommend checking that out if you're interested in learning more. I'll sum them up the best way I can, not really knowing, all the details behind it. But what CyVerse does with these VICE apps is you can customize these apps to have all of the computing needs. They're all customizable via a Docker. So basically everything you need is in the app, so that all students need to have is a stable Wi-Fi connection and a free CyVerse account. Educators and students all access the app in CyVerse, and then everybody has the same resources no matter what machine you're using. So it makes the activity completely seamless. Another sort of intangible that we didn't realize until everybody went virtual last year due to the pandemic. We started out doing this in class. Everybody was using their own computers, but in class we were all together. That immediately shifted to we were all over the place in different states and in different towns using different machines. This worked beautifully. So again, students were all accessing the same computer capabilities within the CyVerse infrastructure, and everybody was able to see the same thing, get the same outputs, have the same computing resources, regardless of what machine they were using. So that's been a huge advantage. So when we were setting this up, my collaborator, Jason Williams of CyVerse and DNA Learning Center fame, he was absolutely instrumental in helping to set this VICE app up. And what we did is we took this FastQE lesson and we put it into a CyVerse Jupyter notebook. So this was my first time using a Jupyter notebook. I had heard a lot about them, and it works really well. We have kind of all of these different prompts that we're prompting the students exactly what to do. Students can actually write into the Jupyter notebook. They can run all of their code in the Jupyter notebook. It doesn't matter if they're using a PC or a Mac. Everybody is sort of getting the same virtual experience by virtue of using the same app. Okay, so here's the live demo part. So we'll see if this works out. I'm gonna stop my share and then I'm gonna reshare. I should, let's see. Okay, I'm gonna reshare. So you guys are seeing my screen, hopefully. All right, so this is the CyVerse discovery environment. I won't go through all the login stuff. I'm guessing many of you know how to do that. But we have 3 tabs here: data, analyses, and apps. We're gonna go into apps and we're gonna do a search for Jupyter FastQE, and that app should pop open. Let's see if it does. Here it is, Jupyter FastQE. Click that to open it. That will open this sort of default launch screen. Just leave all the settings as default and hit this launch analysis. I've already done that because it takes a few minutes to open. What you'll get is a little notification up here saying the analysis was submitted, and then a few minutes later, or maybe even a few seconds later, you'll get this second notification saying your analysis is now accessible through this link. So you go ahead and click the link, and that will open the notebook. It may take a few minutes to open. Again, I've previously opened this so I can have it ready for everybody. And this is what you'll see. This is the notebook opens. And I should say the instructions for how to do all these steps are in the course source paper. So you can refer to that as like a step-by-step. You go into this over here on the left side, you go into this notebooks and you click the FastQE notebook. I'm just gonna make things a little bit bigger. Hopefully that's big enough for everyone to see. Okay, and then in your main screen, you have kind of the directions and background information for the activity. Over here on the left, you have your sort of your directory where everything is gonna go. And it gives you a little background on the experiment. So essentially what we're gonna do is we're gonna take 3 FastQ files from a metagenomics case study that we do in my genomics class, and we're gonna run them through the FastQE. So this gets back to the idea of breaking things off into little bits. We're able to teach genetics students a little bit about metagenomics. They're not actually gonna do anything else with this metagenomics experiment, but we can tell them about this project we're working on. Here's my colleague, Dr. Rocky Parker. He's interested in sexually dimorphic microbial populations on garter snakes. So students in my genomics class do sterile saline swabs of the snakes, male and female. They extract the DNA. They set up 16S PCRs, and then we send all those amplicons off for Illumina sequencing. Now my genomics students, they get that data back and they run it through DNA Subway Purple Line. Students in the genetics lab are just going to use a few of those files from the metagenomics project to run FastQE. But like I said, it does give them a little taste of, here's what a metagenomics experiment is. Here's a little something about that pipeline. You're going to do the very first part of the pipeline using FastQE. And the way that these Jupyter notebooks work inside of the app is, we have all of these instructions and prompts for students. Everywhere where you see a gray box, those are the prompts where students are going to put in their own script. And we have kind of a mixture in here. Some of the script we give them examples. So for example, we ask them first to type in this pwd command for print working directory. So you can either hit the blue bar or control enter. And that command will just simply print out the working directory you're in. So we're in the home/joyvan/notebooks. So that's kind of reflected of what's over here. The first thing we need to do is we need to change directories because we need to get into this data directory to do all of our analysis. So the code for that is going to be cd change directory. And then I'm going to cheat. I'm going to copy and paste, which that's fine if students do. So we're changing our directory from this joyvan/notebooks to this joyvan/data. I'm going to run this command. That's an example of a command where I ran the command and you didn't see anything happen on your screen, but I promise you something happened in the background. Changed our working directory. The way that you can have students see that is rerun the print working directory command. And you can see that they now are in this, they're in this data directory. Let's go in there because right now that data directory is empty. But our next command, we're going to use this very simple command called wget. And that command is for grabbing something off the internet and putting it in your directory. And in this case, what we want to do is we want to grab this, this FastQ, this compressed FastQ file, this fastq.zip file. So the command is wget -o space fastq.zip. And then you need the URL, which we give to them, of where that file is living on the internet in CyVerse. And so this is kind of, we're having students piece together these simple scripts and watch what happens. So I'm going to run this command. And what you'll see is something's happening down here, but watch over here on the left, we'll see that file has been retrieved from the internet and dumped into our data folder. So now we have this compressed folder. Inside that compressed folder, there's 3 FastQ files. So the very next thing we're going to do is use this unzip command. And you can kind of see the process here. We're kind of walking students very gently through. Here are the commands you use to do these very simple tasks. We're going to unzip is the command, and then space. The file that we're unzipping is called fastq.zip. We'll see we're inflating these files and watch what happens over here. We're going to see those files then appear in our directory. So, we're having, and this is like, students who've never done this before find this fascinating. Like, wow, I'm typing in these commands and things are happening. So now we have these 3 FastQ files from our metagenomics experiment. And this is where we can now start to, well, first, if you want to go one step further, you can have them list out their directory using the ls command, it's a common command. So that's just listing out everything that's in your directory. But now at step 4, they're going to actually run FastQE. So the program is called FastQE space. What do we want to run it on? We want to run FastQE on these 3 FastQ files. So our options are, we can type out the names of these 3 files, or we can use something called the wildcard command, which is just an asterisk, followed by a file name, a file extension. So what this command is saying is, please run FastQE on anything in my folder that has a .fastq extension. So that'll run this on all 3 files. It's a nice little shortcut. And so now the program is running. It looks like nothing's happening. This is actually one of the main problems we have is students freak out when they don't see anything immediately happening. It takes about 15 to 20 seconds per FastQ file. Because remember, these have about 10,000 sequencing reads in them. And so they're all being analyzed, and this takes a little bit. So here's where the emojis pop up. Students get really excited. Here's their first file that pops up. You can see it has a lot of high quality bases, but then some low quality stuff at the end. Hopefully we'll get our other two. Right, so here's our second file. You can see it's a little bit lower quality, and then our third file is pretty high quality. So hopefully you kind of get the idea here. We have students run through a few more steps. So they look at a few things in the software help menu. They can see the scale, what each emoji, like what does this smiley face equate to? What does this frowny face equate to in terms of our Fred score? And then the last part of this is they will actually run this FAST-P software, which is the research grade software. And then you could also see that throughout this activity, we have these questions, these sort of activity questions sprinkled in. So they're analyzing the data, they're answering questions. So they have to actually look at their results that they're producing to answer questions. And then you can click right in here, and students can put their answers right in here, and they can save and submit this notebook to you, or this can work seamlessly with, let's say you're using Blackboard or Canvas. We usually have students just make like a Canvas quiz or something like that. So that's the extent of it. I mean, it's a really easy, I won't go through the whole activity, but it's a really easy short activity. Students definitely struggle with it. They get errors. They're gonna, they don't realize things like the syntax in the script has to be exact. But it is, I think, a very useful experience for students to go through to see what writing scripts are all about. Like, it can't be close. It's got to be exactly. You either have to have that space there, or you can't have that space there. So it's a pretty fun activity. It's really easy to use. Let me see what else I have to say about it. Okay, so again, here's the core source paper. If you click into the supporting materials, supporting file S6 has the FastQE Jupyter Notebook implementation. So it's just a short Word document that kind of walks you through all those steps for getting the Jupyter Notebook page set up. And then it's very easy to run. All the instructions are actually in that Jupyter Notebook. One thing to be aware of is you need to pre-register to use VICE apps. So that's one of the things that is a little bit different with the VICE apps, is that you have to contact CyVerse support and say, I would like to use this VICE app with my students. And then you need to provide CyVerse usernames and email addresses for everybody that's going to use the VICE app. So that usually happens pretty quickly within 24 to 48 hours. But I would recommend allow at least a week, I would say, before you want to bring this into your class to make sure it's up and running. Okay, and just to sort of summarize, I've been trying to consider barriers to incorporating bioinformatics analysis in class. I've also been thinking about how to effectively push these activities early on in the undergraduate experience and keep stacking the complexity. We can go up and up and up. Students are really resilient. The more you teach them earlier, the higher they're ready to go later. And I've been finding a lot of effectiveness in using pieces of pipelines rather than saying, okay, everybody bear down. We're going to spend the next month learning this really, really meticulous pipeline. And really, just finding engaging ways to teach bioinformatics, I think, is really important. It's not like I'm teaching bioinformatics PhD students. I'm teaching biology undergraduates. They have a diverse set of interests. So you really have to put some effort into engaging them. And I think that's a really worthwhile endeavor. And, the through line to all of this is CyVerse has the tools. They have the people that are ready to help you to do all of these things. And I have just been really fortunate to work with a bunch of really great people at CyVerse, as well as the Cold Spring Harbor DNA Learning Center over the past few years that have really helped me out with all these things. So I do want to plug these workshops once again, because we're looking for participants. So before I end, if you have any interest in DNA barcoding, so Sanger sequencing analysis, or DNA metabarcoding, next-gen sort of microbiome, environmental DNA analysis, please check out this link. Again, I'll put it in the chat in a second when I finish up. We are running a third workshop. We have a lot of workshops this summer. This one is going to be probably a little bit more advanced. It's called Getting Started with R and CyVerse. Jason and my colleague Karen Bernard-Kubo are running this workshop, also virtual. And this is going to focus on if you would like to use R and RStudio analysis in CyVerse using these CyVerse apps, then this would be a good workshop for you guys to check out. So very lastly, I just want to thank a lot of people. First and foremost, I want to thank my genomics students. So this is my fall 2018 genomics class. They were the guinea pigs and also the helpers for taking this FastQE idea that I saw on Twitter and actually developing it into something. Particularly Max Mazza and Rachel St. Jacques, they were kind of above and beyond students that had some scripting background and were able to help me. I'm a complete novice when it comes to scripting. I also want to thank the 240 genetics lab instructors who were kind of varying. They were game for giving this a try. I think some of them were more concerned about scripting in class than others. My JMU collaborators, Ollie Heileman and Kaylin Murray, the co-authors on our course source paper, Max, Rachel, Jason, Kaylin, as well as Sabrina Robertson and Andrew Lonsdale. And then I've been lucky to have a really strong relationship with the Cold Spring Harbor DNA Learning Center over the past few years. Director Dave Mikolos, co-director Bruce Nash, and of course, Jason Williams, who's just like the guru of all of this stuff. So I'm really, really thankful to have these guys as collaborators, as well as all of the CyVerse support staff. They've been really instrumental in helping me with all of this. And with that, I will end and I'll take any questions. I'm also gonna put all the stuff in the chat that I promised. So here's all of my webinar resources. Put them in the chat. So, okay, I've got metabarcoding workshops, the R and CyVerse workshop, a link to my selected works webpage that just has a lot of in-class activities that you can take and try out, course source paper, and then my email and Twitter feed where I have all of this stuff pinned. So I will thank all you for listening and be happy to take any questions.
Wow. Hey, thank you so very much.
Sorry if that was fast, but I saw I was getting late. So
no, this is great, really great. There have been questions, and I think you've covered most of the answers in your talk.
Okay.
So I'll just start real quickly. What was the research question that students were pursuing in the cure, which you?
So in the metabarcoding, the snake cure or the DNA barcoding cure?
Actually, I posted that early on when you were talking about the intro course. I think you should go on to the other questions at this point. I can read your paper. Thank you.
Okay.
Oh, I mean, let me answer it really quickly. If you're talking about the barcoding cure, what we do is we leave it really open-ended. We allow the students to investigate whatever they want using the DNA barcoding pipeline. And that can be investigative stuff, like they go to their sushi restaurant down the street and they see if the fish that's in the sushi is as advertised or if it's some cheap version, or they can do some ecology project where they're looking for different populations and beetles and different kinds of cow dung. So we put it all in the students' lab.
Thank you. That was great.
Okay. Another question, Ray, is which program to use to run command line for students that have Windows? And you mentioned that very challenging problem.
Yeah, so again, I will fully put myself into I hope that this webinar did not make you think that I am some scripting guru. I'm in the novice category, so I don't know. The little bit I know how to do is on, I am a Mac user, so I use Terminal in Mac. I don't think I even know how to open the command line on a PC. So I'm not the person to ask. And I will say, just to plug the CyVerse VICE apps again, you don't even need to understand any of that. So, if you're like me and you're coming at this from a very low level of understanding, the VICE apps make it so that you can open up the Jupyter Notebook and everything is in the Jupyter Notebook. So I do apologize. I wish I had an answer to your question, but I don't even have that much requisite knowledge to say, here's what you do if you have a PC. I can tell you one thing, that the emoji thing will not work on a PC just because Windows does not support the use of emojis as fonts, which is kind of a bummer. Maybe that'll change sometime. So this really only works. You could use this, the emoji activity on a Mac or Linux, without CyVerse, or you can use it in the CyVerse app on any machine. And that's why I think the CyVerse app is the way to go if you're thinking about doing this in class with students.
Okay, thank you. Great course. It would be highly appreciated if there's an imputation to the missed or bad quality data on how to impute them against software or using software against the reference genome. Maybe you can just recommend, I guess, an app to do that.
Can you repeat that? Oh, okay. Would it be highly,
I think they're asking how would you
missing or bad
filter
quality data?
How would you filter the bad data?
Oh, I see. So, well, the FastP software would be one example. There's tons of software. So I'm trying to think of the popular ones. Trim-o-matic is a type of software that specializes in trimming and filtering FASTQ files. A program called FastX. So there's a lot of different things. It really comes down to preference. Say in CyVerse, the app I tend to use in CyVerse is called Trim-o-matic. You can customize exactly how Trim-o-matic is going to filter your reads. And I think what I would do is I'll point you to, if you look in all the resources I posted, there's one that's called, it's a selected work page. And if you look on that link, I have a whole bunch of sort of, these are non-peer reviewed, they're just sort of PDFs of lessons that I use in class. And one of them is trimming and filtering FASTQ files, and we use Trim-o-matic for that.
Okay, another question asks about seeing or visualizing the quality control with box plots of the green, yellow, and red zones. And I think you said that that was the subsequent part of the Jupyter notebook, right? Using FastP, was it?
Is it this question? Is it possible to see QC with the box plots of green, yellow? Yeah, so the green, yellow, red, those are the FastQC plots. So remember, FastQC is kind of the standard way the genomics community assesses the quality, and FastQE is the fun emoji way. So I would say if you want sort of research grade QC, I would recommend the program called FastQC. FastQC, and yeah, that's a I want to say one of the most widely used softwares in NextGen sequencing analysis. And you can run that command line. You can sort of run that command line on your computer. The way we usually run it with my students is CyVerse has a FastQC app that is just one of the easiest apps to run. So I like to run it in CyVerse discovery environment.
I'm gonna have to send you that check and pump it up a little, Ray. You're promoting us so beautifully.
Yeah, yeah. Hopefully the check's in the mail.
All right. Let's see a few more questions if you have a couple minutes or some comments.
Absolutely.
Somebody says you can use Ubuntu in Windows. And can someone access all the teaching stuff or just learning it themselves? Meaning they don't have a class to teach. Yes, of course, right?
Well, yeah, of course. I will say for the workshops we're advertising, we have applications for those. What we're really looking for is a commitment for undergraduate educators that are committed to bringing their stuff into class. But I would say for someone that doesn't necessarily have a class, but just wants to learn, check out all of those tools that are on the works cited page or the course source paper. Yeah, of course. I mean, it's always good to just learn. I see a question about workshops being offered next summer. I do not know. We were actually only going to offer 2 summers of workshops, but since everything has gone virtual, we're no longer like flying people all over the country and paying for hotel rooms. So at this point, we're doing workshops this summer, even though we weren't planning to. I have a feeling we will next summer just because it's virtually cost-free to do virtual workshops. But I don't know. All I can say is we're definitely doing the barcoding workshops this summer. We do tend to have a JMU workshop every summer, but as far as what the content will be next summer, I have no idea.
Okay, so we're getting up close to the end of our time, and I'd like to thank Ray so very much for sharing this really fun exercise. Even novices, I think, can basically grasp the process as well as just not be intimidated and be able to take next steps. So thank you so much. CyVerse webinars will be on hiatus for this summer, and then we'll resume in the fall. We're working on a great lineup to showcase many of the new features and capabilities that we'll be rolling out this summer, all of which are designed to improve your ability to do collaborative computational research. So until then, everybody have a great summer, and thank you so very much, Ray. Yes, we will be posting this video on our website. So we'll see everybody back here at the end of summer in August.
Great, thank you so much. Thank you for having me come.
Thank you. Bye now.